Guys, welcome to Module 4. This is by far my favorite module to teach because this is where we take all of the knowledge and information that we collected over the last 3 modules and we kind of put it into an application, right? We kind of see where it all leads to in terms of biology. And so we get a good chance to check out how genotypes ultimately affect phenotypes. We get to talk about how phenotypes are acted upon by natural selection and evolution and all those mechanisms. And then at the very end of this module is sort of a treat. I give you the history of life on this planet. Now, obviously, that's a boiled down, like very condensed version of 4 billion years of evolution. But it's nice because then you kind of get to see like the process. You're like, okay, this is how everything works in order, or how everything kind of like showed up on this planet and in what order. The reason why I do that is just because when I was a student, I never had anybody like go through that with me. I didn't take a geology class or anything like that. And so I thought maybe that was one of the things I missed out on. And so I just wanted to provide that for you guys. So anyways, just something else just to keep in mind too, is that online. So not only is this a summer course, this is an online biology course. So there's no in-person lectures. So in reality, this course is actually like more than twice as hard as you would normally expect from a regular semester. So if you're like, man, those exam questions are really tough, or those lecture quizzes questions are really tough. The reason why I do that is because it's open note. I can't control what you guys have in front of you, and so I have to write the questions in such a way that if you understand the concept, you can understand the question. As opposed to like when I do in-person classes, I usually give them like a study guide or something like that that has almost all the test questions on it. Like, maybe they're worded just slightly different, but they're literally in order of the test. And that way, because they have to memorize those, they come in, they see the test, they get the questions in the order they're in. For this stuff, like I give you more content, so I'm not constrained by like, oh, we only have an hour and 15 minutes in lecture. I give you more content, and then and so that's makes it difficult. The questions on the exams and quizzes are more difficult, and then also like the amount of information that we cover is more difficult, and then also like the amount of time we do it in is shortened in half. So if this feels like a lot like a rushed course, well, that's probably why is you're taking in a lot more than you normally would if it was just a regular semester long in-person class. Okay, so having said that, that's my caveat. Welcome to Module 4. Let's get started. So last unit, we talked a little bit about DNA replication, which is purely used for cell replication. So anytime you duplicate your DNA, you're obviously going to go through a cell division, so you end up back in a diploid phase. Unless, of course, you're making sperm and egg, and then you go through another round of divisions, and you end up with haploid sperm and egg. But anyways, we talked about replication, and then we also talked about RNA transcription. So those are two totally separate things. RNA transcription is like, okay, you have a sequence of nucleotides in your genes. How do we get that sequence of nucleotides and turn it into a sequence of amino acids? And that's RNA transcription and RNA translation into proteins. We touched at the end of that about mutation and how mutation works and how it can like lead to a change in protein structure. And the change in protein structure would be a change in the amino acid sequence, which would result in a change in how that protein actually functions. So what we're going to do next is we're going to kind of widen out those slends and we're going to look at the big picture. Okay. So most animals and plants as well, but plants are a little bit like more tricky than animals because a lot of plants actually, instead of being like diploid, they're like polyploid. So instead of having 2 sets of every chromosomes, some plants have 16 sets of every chromosome, and that gets like super confusing, especially if you're trying to do Mendelian genetics with them. Okay, I digress. So most animals that use sexual reproduction, most of them do, will cycle between the diploid phase, which is where we're at here, and this would just be like a regular individual, and the haploid phase. And so the diploid phase, you got 2 copies of every gene. The haploid phase, though, would be something like if you're making sperm or egg. And so they cycle between a diploid phase, like I'm in my diploid phase, you're in your diploid phase. But when you go to reproduce and you make a gamete, or you make a gamete like sperm or egg, that gamete is haploid, and that's you. So you're going through a haploid cycle. Maybe your consciousness isn't traveling with it, maybe it is, but you're going through a haploid phase that will then become the diploid phase for the next generation. And so when an individual gets ready to reproduce, they take their diploid phase, their genetic material, they exchange alleles between homologous chromosomes, and then they go through a round of division, and they end up with half of the amount of chromosomes that they started with. So if you're diploid and you go through a division, you end up with half the chromosomes, you're haploid. You only have one set of chromosomes. And so that way, when the haploid sperm and the haploid egg meet, so each with their own set, they come together and make a diploid zygote. And I have a little cool animation here. Just so you know, I'm only using just this third chromosome. Ideally, if I had enough space, I would include every single 23 of these chromosomes. But really, just imagine that there's one of each inside of the sperm. And right now, it's just this one, right? And then you have an egg, which is a lot bigger than a sperm, and it's got its own set of chromosomes. And they come together and they make a fertilized egg or a zygote, which will be an embryo and go through development. All right. So when an organism's in its diploid phase, it's going to have two copies of the same chromosome, right? It's going to have two copies of the same chromosome for each of the 23 pairs of chromosomes. So you got 23 pairs of chromosomes. The two copies are identical in the order in which genes appear on them. Okay, so check this out. So on this chromosome on the left, that would be like the paternally inherited one from your father. And then, so he's got a gene for eye color. He's got a gene for height a little later on the chromosome. And maybe down at the last part of the chromosome, he's got a gene for hexokinase, the enzyme that phosphorylates glucose in glycolysis. So anyways, we have three genes. Obviously, there's way more than that on this chromosome. And notice that the order and sequence of genes are the same, right? So gene 1 is eye color. So the chromosome you got from mom has a gene for eye color. It's going to have a gene for how tall you are. It's going to have a gene for your hexokinase. But the difference is that they're not identical when it comes to the alleles. And I put the alleles in parentheses. So alleles are different variations of those genes. So you have the exact same order and sequence of genes, but the alleles can be different. Okay, so the variations of the gene. All right, so let's say that we have, some region of DNA that looks like this, right? So you got probably template strand on top, you got your sense strand on the bottom, and maybe it's a gene. Okay, so it looks like that, and we'll call it a gene, sure. And it comes from that chromosome, right? Some order and sequence of nucleotides. Now remember, for every gene, you're going to have two copies, so you get one from each parent. So all I've done is I've duplicated this copy, and so what this is showing me is that I got one of these copies from my paternally inherited chromosome, one of these copies of the gene from my maternally inherited chromosome, and they're identical. They're exactly the same. And so if they are the same copy of the gene, we say that they are homozygous. They're two identical copies of the same gene, meaning the gene sequence is exactly the same, the sequence of nucleotides. Now, if we come down here and we see that we have the same gene, right, on our paternally inherited chromosome. Now consider that on the other chromosome, there was a mutation. So we have paternally inherited chromosome, that sequence is reading just like it was on the last page. But the maternally inherited, I'm gonna switch to my pointer. But the maternally inherited chromosome has a mutation on it in that gene. And so the order and sequence of nucleotides are slightly different between the two genes. And so what do we say that is now? We say that individual is heterozygous, meaning different zygosity or different alleles at that particular gene locus. Now, I'm going to give you a caveat down here, right? If the gene or if the mutation leads to the same amino acid being produced, so think maybe if it's in the third position, which is a little more flexible, that codon might be different. It might be like, in this case, what is it? It's like instead of CAT, it's CGT. And maybe that still codes for the same amino acid. And so if it codes for the same amino acid, that means the proteins are going to be identical. So even though there's two different sequences, if the proteins are identical, we're still going to say that's a homozygous individual. So in order for them to be heterozygous, it cannot be a silent mutation. Okay. Moving on. So we've got our paternally inherited chromosome, got our gene, And we'll call this variation of the gene big B. So we'll just give it a designation, right? So a lot of times you'll see this where we use a capital letter to designate maybe a dominant gene, or a lowercase to represent a recessive gene. But we're going to call this variation of the gene big B. Typically, when you see the capital letter, that's a wild type variation, meaning if you were to go out in the population and sample 100 people, 90 of them are going to have this copy, at least one of these copies of the gene. 90 out of 100. So, and I'm just making that number up, but it's going to be a lot of the people that you sample in the wild, or the animals. If you go out and sample a bunch of, I don't know, like rodents in your community, and for a certain gene, they're all going to, most of them are going to have the same variation of that gene, right? We call that wild type. And so typically the wild type, what you find out in the wild is given the big B designation. But now in the case of our mutation, we have to say, we have to designate this sequence of nucleotides as something different than this sequence of nucleotides, because they're two different sequences of nucleotides. It's the same gene, but we have to say, okay, this variation of the gene is big B, this variation of the G, we'll call it little b. And this would be like our mutant variation. So this is like maybe the recessive one. We don't know yet what its function is, right? Because we're speaking in general terms, but typically mutations are not beneficial to organisms. Usually they result in loss of functions, right? If you just randomly change a nucleotide in the sequence and change the amino acid in the sequence, that changes how the protein folds, changes how it functions. Usually that protein is pretty optimized for its function, and any changes in this amino acid sequence are going to make it less functional. And so we call that more of like a recessive type. Yeah. So if they're two different sequences, right, we'll say they're heterozygous, and we already knew that from before, right? Okay, so we have basically this individual is heterozygous, which means heterozygous individuals have two different copies of the same gene. The difference between the two copies was likely a result of a mutation that could have happened generations ago in an ancestor, right? It just keeps getting passed on. All right, so we've got our B allele from our paternally inherited from our father chromosome, our little b allele from our maternally inherited chromosome. The genotype of this individual is heterozygous. And the genotype, what we mean by that is we just say the genotype of an individual is a particular set of alleles that that individual carries. So if the individual had two copies of the big B, their genotype would be big B, big B. Genotypes can be even bigger. We can expand the genotype and include more genes. So you can have a genotype like big A, big B, little c, big D, little d. You can have a huge genotype. But if we're just talking about one gene, we typically only give it in like, okay, you're big A, or you're little a, little a. And for the most part, when we get into Mendelian genetics, we'll do two at the most, two genes. So you'll have like big B, little c, little c. That's the most that we'll do in this course. When you get into genetics, you can do like 3 genes, 4 genes. If you're into linear algebra, you can do like 20 genes. It gets kind of crazy, but yeah, don't worry about that. Anyways, okay. So genotype to phenotype. We've got the genotype big B. That's saying that that particular sequence of nucleotides codes for, you know, the big B product. And this particular sequence of nucleotides that's slightly different on the other chromosome codes for a slightly different product. And we might say, okay, well, the big B codes for black hair. And this is not how hair color in humans works. This is just an example. So big B codes for black hair. Little b is going to code for blonde hair. But then that begs the question, right? Well, what if the genotype is big B, little b? What if they're heterozygous? What kind of phenotype are we going to get? Is it going to be white and black stripes? Is it going to be just black? Is it going to be gray? Who knows? I don't know. So we know big B codes for black hair. We know that little b codes for blonde hair. So if we see only black hair in the phenotype, we call that complete dominance. So this is where one allele masks the effect of the other allele. We can say that it's completely dominant, complete dominance, right? So you can say completely dominant or complete dominance. They're the same thing. And I give you the example, one parent has black hair, the other has blonde hair, and all their offspring have black hair. That's complete dominance. All right. So what if the heterozygous genotype leads to both alleles being expressed discretely in the phenotype? And by discretely, I mean like, okay, I can see that allele is coding for black hair, that allele is coding for blonde hair, and they're expressed in the same individual by themselves, separate from one another. We see this in blood type a lot. So blood type would be an example where one parent has A type blood, the other one has B type blood, and the offspring has AB blood. They don't have a blend of AB, they just have A and B. And the reason why is one parent is going to have an A receptor on the red blood cells that's coded for by an allele. The other parent is going to have a B receptor on their red blood cell, and their offspring are literally going to be like this lady over here. Is they're going to have an A receptor and a B receptor on the red blood cell, not a blend of the two. They're just going to each have their own receptor. O type has no receptors, and so you can have a parent that has an allele for an A receptor on their red blood cell, and they also have an O allele. So they have an A allele and an O allele. Well, the O allele doesn't code for anything. It's just like null. So that individual that has an A allele and an O allele will always just express the A. An individual that has two O alleles won't express any receptor on the red blood cell. The O's are null. They don't make anything. And since you have two copies of every allele, you could have a B allele and you could have an O allele. And in that case, you would have B blood type. You could have a B allele and an A allele, and you would have AB blood type. So that's how blood type works. It's a codominance thing. You also see it in spots on a cow, right? So there's parts of the cow where you have like white spots and black spots. Each of the alleles that are coding for those hair colors are being expressed with codominance at different parts of the cow. All right, moving on. What if the individual has like silver hair? So we have the black and the blonde seem to blend together and to make an intermediate phenotype. And we talk about this a lot when we get to selection, natural selection. Because what this means is there's three phenotypes for the three genotypes, right? And that's the same way with codominance too. There's three phenotypes. Think about this. Okay. If you have big B, you would have black hair. If you have big B, you would have gray hair. If you have little b, you would have blonde hair. So you get the three phenotypes matching up to the three genotypes. An example of incomplete dominance. So this is where the alleles blend together in the phenotype. An example would be sickle cell anemia. And you see this when we get into selection and stuff for the heterozygous advantage. It might be that that intermediate phenotype, that gray-haired person, has higher fitness than the black hair and the blonde hair. It could be. And what that would mean is, since this individual, think about this, this individual is carrying around one allele from each. If they do better in the population, like if they do better, they reproduce better, they find more food, whatever it is that phenotype, right, that intermediate phenotype is providing them, or whatever benefit it's providing them, right, they're going to leave behind more copies of both the big B and the little b in the population. And so what this does is it stabilizes those two alleles in the population because the intermediates have higher fitness. And we'll talk about, I'm jumping way ahead here. Continue. All right, so that's incomplete dominance. And then finally, right, well, this isn't the heterozygous individual, this is just the homozygous recessive. So anytime you have two, or you're homozygous for the recessive allele, and you have a unique phenotype with that genotype, we call that recessive. So this is when the heterozygous has, when the homozygous individual, so the two recessive alleles, when they're paired together and we see a unique phenotype, we call that recessive. An example of this would be like any sort of recessive disease that you've seen a lot of. So maybe you haven't seen a lot of these, but they're familiar to you. Hemophilia. Hemophilia is a recessive blood disorder where they don't make platelets very well. So it appeared in the British royal family. So what's really interesting about royalty is royalty in the past was, sort of this like, oh, they've got specialness. They talked to God or whatever it was. And the people that were in the royal family were considered all sort of, they had elements of the divine within them. And so in order to keep that within the family, they would inbreed with each other. The problem with inbreeding within a family is that you end up getting these recessive alleles, right? If they show up in the family, they stay in the family and then they duplicate and multiply and they get paired together more often. And so hemophilia is actually a disorder, a blood disorder that ultimately came from England royalty. Is that it became very common for children in the royal family to have hemophilia where they couldn't clot, they would just bleed out. And you could see how that would look like sort of witchcraft or like someone had cursed the family. So you see where some of these things come from. Albinoism is another example. So that's where you don't make pigment in your skin, hair, nails, anything. Cystic fibrosis is another one. That's where you do not make good surfactant on your lungs. So that means that your alveoli in your lungs are not able to expand fully. You have a hard time getting enough air, and it's sort of like suffocation. It's really an awful disease and very painful. Yeah. And that's the end of this lecture. I will see you guys for the next one. Bye.